Ladies and gentlemen, I present to you the 2013 Bob Pinedo Prize recipient, Professor Patty Johnston.
So, Marty, thank you, and thank you, everybody. I know you've been here and all had a long day. Some of you are real friends. Some of you have collaborated with a long time ago. I see Fritz there at the back of the hall, who worked with me a long time ago on thymidylate synthase. And Bob has been a real inspiration to me, and I'll come to that now in just a minute, along with Bruce Chabner. My title that I've chosen is Translational Cancer Medicine, A Journey from Concept to Reality, a lot of which we've been listening to today, but also a translational journey for me personally from Bethesda to Belfast, because a lot of the principles that Bruce taught me and others within NIH back in the 1980s and 1990s really very much stay with me today in terms of both how I treat cancer patients, but also how I approach this whole problem in trying to tackle it. I first of all should just declare my conflicts of interest, founder of Almac Diagnostics, Fusion Antibodies, and then where my research funding and advisory functions come in. My dear wife, Iseult, I certainly would not be standing here. Marty, as you know, she refers to me as a work in progress. It certainly is that. I have my moments, as we all do. But Iseult, I wouldn't be standing here, nor would I have done any of the things that Marty has talked about if it hadn't been for your wonderful support over many, many years, and long may that continue. This gentleman, whom in essence we're all honouring, I met Bob for the first time way back in the early 1990s, actually here in Amsterdam at the old NCI EURTC meetings that we used to have every 2 years that he and Bruce used to organise. Those used to be fantastic meetings on drug development. It was a real honour to be here 8 years ago and to see my mentor receive and be the recipient of the very first prize, Bruce Chabner, because, well, personally for me it was a real delight to see that for a whole host of reasons, but also because Bruce to me emulated the whole spirit of what Bob Pinedo was about in terms of cancer patient care, whether that was directly with patients, research or beyond that. And indeed all of the recipients that have gone before me have very much been in exactly that same mode. So I'm very honoured to be standing here as the 8th recipient, indeed humbled, and I hope I can live up to the legacy that the others who've been recipients before me, and I'm sure will come after me, already have done. So where did this translational journey begin? Well, it began in Bethesda back in 1987 when I received a fellowship in medical oncology at the intramural program, working in Building 10. I remember the first day I went to this building, I actually came off the metro, and I just kept walking and walking and walking. And then this edifice appeared in front of me. I said, that's huge. And I felt I was walking through a village. Remember, I came from a fairly big city, Dublin. But when you walked onto that NIH campus for the first time, I have to say it really blew my mind. And I'll never forget going, because I went in the back door. This isn't the front door of Building 10, it's the back door. And I'll never just forget that the impact, just that walk from the station to this building. And I have to say, the minute I walked in that building, the bottom line was I never looked back. And that's the absolute truth. Here are a group of fellows who were fellows with me. Eddie Chu and I worked together for many years in the lab. Unfortunately for this group, there were 2 Irishmen in it, Frank Sullivan and myself. And that's why we were known as the Friday Afternoon Club. Believe me, it went along well after Friday afternoon. But we had great fun as fellows together. Philip Browning, unfortunately, died about 8 years ago. Iseult and I were very friendly with he and his family. He died, unfortunately, from colorectal cancer. But we really had a great time as fellows, working very hard. And all of them, Stan Lipkowitz is still there at NIH, right over here in the corner. All of them have gone on either to continue in the research field or to work in industry. Frank Sullivan is currently Professor of Radiation Oncology in the University of Galway. And here were the tough guys, Eli and Bruce. We had many mentors We really did have many mentors as fellows, but these two guys were special, and for very similar reasons. They cared about the fellows, they really cared about the patients, and they really mentored you. Going on ward rounds, on Friday morning ward rounds with these two guys was like opening up an encyclopedia of what's what in cancer. And it really was that sort of really, as a fellow, allowed you to start at chapter 1 and actually feel like you were already at chapter 10, because you were digging into this depth of knowledge that in essence, these guys and others around, Vince DeVito was still there, Mark Lippman was still there, Bob Young was still there. So you were immersed around people who really had been at the forefront of oncology from some of its earliest days. But very importantly for me, in terms of then, was they also brought a philosophy which was about accelerating cancer discovery through integrated clinical and scientific research to improve the outcomes for cancer patients. And that was something that was writ large within that whole culture, that clinical culture. Everybody was there, was on a research protocol. We as fellows were writing research protocols. But importantly, we were trying to generate ideas coming from the lab programs that actually would move ahead. And this was 1987, 1988. We're not talking about 2004 or 2005. So this really was translational research at the germinal stage, because back then we didn't have any of the technologies that we largely take for granted today. So when I went into the lab, I went to work with Bruce and with Carmen Allegra. And the challenge I was given was actually, how do we create a personalized medicine strategy around fluorouracil? That was really the challenge that was given to me. Now, contextualize this. There only was fluorouracil. Platinum had been there about 10 years prior to that. But we didn't have irinotecan, we didn't have oxaliplatin, we certainly didn't have biological therapies. And indeed, Charlie Heidelberger had invented 5-FU, back in 1958. Peter Dannenberg, who was part of that whole operation, then became a very close collaborator, still is somebody that I have worked very closely with. And indeed, Fritz, at that time here, was working also on schedules and how to actually give fluoropyrimidines better. And one of the things that I was tasked to do was to begin to build assays around how to quantify this enzyme, which was the target for fluoropyrimidines. Indeed, 5-FU, you could have argued, was actually a targeted therapy, because its major action really was to inhibit this enzyme by forming a stable complex in conjunction with reduced folates, and thus actually starve DNA of thymidine precursors, so that you actually had uracil misincorporation or other problems fracturing the DNA. And that's what I did. I actually developed the first monoclonal antibodies. That took me 2 years. Now to make a monoclonal antibody would take me 6 weeks. But that took 2 years of really hard graft, and then a following year of validating all of the antibodies. There were actually 8 antibodies in total from 8 separate hybridomas that I actually made. Using these, I actually showed that the level of thymidylate synthase, both activity and also protein, was directly related to the sensitivity of fluoropyrimidines. We then started to do that in tumor biopsy samples from people with breast cancer, colon cancer, gastric cancer, head and neck cancer, pre and post, measuring both free enzyme and complexed enzyme. And went on to basically show that the level of the enzyme, if it was very high, that you tended to have no response to fluoropyrimidines, whereas if it was low, you tended to respond. This is something that I did in collaboration with Peter Dannenberg, where Peter was developing PCR assays, and we combined our labs to actually look at a whole range of these markers involved in pyrimidine nucleotide synthesis or degradation. Fritz was also involved in this as well. And one of the consistent findings in the metastatic context was that if you have high levels of TS, and that still is true today, and let's not forget those lessons, don't be giving people fluoropyrimidines. They are highly unlikely to actually respond. Importantly, we also were able to show that TS in the metastatic context, and I'll come to the adjuvant context, was also related to survival. And again, this is just with single agent 5-FU. If you had very high levels of TS, you actually tended not to respond and die very quickly. Whereas if you had higher levels, if you had lower levels, you actually had a median response that went out somewhere to 12 to 14 months. So you actually tended to do much better. One of the real surprises, however, was when we did some of the big adjuvant studies with the NSABP initially. This is the original study that was done looking at TS in the primaries from rectal patients. And what we found was that TS appeared to be a prognostic marker. And that prognosis appeared to be independent of pathology issues, both around stage 2 and stage 3. This is the stage 3 data set. So low patients tending to do well, high patients tending to do much worse. And so we not only had a marker that was predictive in the metastatic context, we had a confusion in that this was picking out some biology that even today we probably don't fully understand. One other thing that we never published was that there was a subset of patients within the adjuvant setting, based on TS levels, that we were actually harming, particularly in stage 3 disease, who, when we were giving them either MOF chemotherapy or 5-FU-leucovorin, appeared to do worse. And those in particular were a group of patients who had low TS. But we were never able to get that to statistical significance, but I think it's an important vignette in that I still believe, and some of the things that Emil was talking about today I think resonate in that direction as well, that there are a subgroup of patients in there that we actually are harming, and let's try and find out who they are. Now, what were the key lessons here for me while I was at NCI? Well, first of all, the importance of personal passion and commitment to what you're doing. There's no substitute for that. The second was the importance of a dynamic and stimulating training environment. Because I'm nothing special, but getting into this environment was a massive opportunity for me to really develop myself, and I used every sinew to actually make that happen. One of the really important things, however, was the importance of developing a highly analytical and challenging approach to medical decision process. So that you didn't just come and take the textbook out and say, that's the way it is. You also didn't come to Paddy Johnston or Marty Murphy and treat them exactly the same. Each patient was different, and it was beginning to take that sort of feedback loop of let's think carefully about what it is we're doing, why we're doing it, and let's learn from every patient. The other thing was about the importance of mentorship networks and the development of leadership skills. It was very clear to me, even as a fellow, that the people who were really impacting were those who were standing up, thinking carefully, but also making bold statements about what it is we were and were not doing. And then the importance of integrating that clinical and scientific research to improve the outcomes for cancer patients was really, really pivotal. And then finally, the importance of teamwork in all of this. I think everything that you've heard today, none of it would be possible if you went and tried to achieve it on your own. You have to, as Tim Maughan's talk has highlighted very clearly, to achieve something like what he's trying to do with Focus 4 or Focus 3, you need a whole network of people and patients who are going to work with you to achieve that. But also the importance of all of this in developing friendship and kinship. Because the people that you collaborate with, that you work with, you'll work with for a lifetime. And it's in working with people that you'll actually learn both about them, who they are as people, professionals, but also together what you can actually do to improve the lot of patients. In 1996, I then took that philosophy back to Belfast. Bruce tried to stop me going, and he was absolutely right too, because it was probably the gutsiest decision, but also the second best decision I've ever made. Well, actually, no, the best decision I've ever made was to marry the woman who's sitting up there. Going to the NCI and going to Belfast were the two best career decisions I've ever made. The reason why, there was an opportunity and a challenge, and I grasped them. The challenge here was to really bring modern cancer care into an arena where there were very intelligent people, excite them, get them motivated, and start developing that ethos of clinical science and translational science together. This is one of the first conferences I ever held. Richard's sitting up at the back, he probably remembers it, where there's Henry Lynch, David Lane, Dan Haller, Heinz Joseph Lenz, Peter Drake, or Peter O'Dwyer, excuse me, Elaine, and Richard Wilson, who at that time was I think, just about to go to the NCI to work for 2 years. And all of these people and many others actually came around the development of this project. This actually was a model of the cancer centre that we were building and that we had just started. Now, the important thing was that it wasn't just about building a building. Along came the 3 Irish tenors. This is Quentin Coey, who was the chief executive of Belfast City Hospital. And when he hired me, he had no idea what he got himself into, because here was Professor Murphy on the left, and here's Tom Moran, Chief Executive of Mutual of America, on his right. Bob also played a very important role here, because I brought a whole TV crew to Amsterdam. Fritz, you may remember this. And the reason I did that was I wanted them to see what Bob had done at the VRU, because to me that was exactly where we should be headed. And so, guess what? They actually did a documentary with Bob and other people from here, appearing on that, but demonstrating the importance of this type of integrated clinical care for cancer patients. The importance of these 2 guys was Marty, well, I'll come to this later. Marty basically walked every step of this journey, and believe me, there were many very, very tough steps along the way. Bruce walked every step of the journey, taking some of the fellows over to MGH, the Farber, and beginning to actually allow me to spot the good guys and say, off you go and get trained in great centres. And Tom actually, very importantly, brought that quiet political support, that external affirmation that says there's something higher going on here. And indeed, Marty was very much in that mould too. Here was the other man for all seasons. This is someone none of you know, well, Marty knows or knew, and Anne knew very well. This was Sir Alan MacLay, who unfortunately died just 3 years ago, and is a huge loss to Northern Ireland. Alan was an entrepreneur par excellence, the founder of Galen Pharmaceuticals, and also of Almac Sciences, or the Almac Group. We'll come to it later, but when Paul Harkin and I came up with this idea of developing disease-specific arrays for use in paraffin, but also as tools for discovery, Alan immediately came in behind us. We had formed a small company called Aradex at the time, and Alan came in not because of who we were, but because he wanted to really make an impact, and boy, is he making an impact, even though he isn't here to see it today. He also, of course, again in the same mode as Marty, Bruce, and also Tom Moran, became very important in pushing at the politics that goes on behind getting people moving in a society in a coordinated direction. And he's badly missed by all of us. So this is a very famous photograph. This is the signing of the NCI All-Ireland Cancer Accord. This happened in 1 year after the Good Friday Agreement. As Marty said, there are only 2 things related to health in the Good Friday Agreement. One is in cancer, the other is in ambulance cross-border services. So it was quite a feat to actually get that in there. That actually resulted in this very unique accord between the National Cancer Institute, Department of Health Human Services, and Departments of Health Northern Ireland and Southern Ireland, and led to huge training opportunities for doctors, nurses, scientists, which goes on to this day, to go to the NCI or comprehensive cancer centres funded by the governments north and south in a way that actually has really captured the imagination and indeed has now been replicated in 4 other countries. This is Ed Liu, who went on to run GIS in Singapore, Rick Klausner, who at that time was the director of the NCI and whom I knew well when I was there. Harold Varmus at that time was the director of NIH. And of course, this is Etta Campbell, who was our chief medical officer. Brian Cowan, who became Prime Minister, he was of the Republic of Ireland. He, at that time, was the Minister of Health. Etta Campbell was our chief medical officer, and Senator George Mitchell, who became a very good friend, was also Chancellor of our university. And George heralded this as one of the really important things that he presided over during very difficult days, negotiating the lasting peace that now is, thankfully, in Ireland and in Northern Ireland. So what did all of that lead to? Well, this is now buildings again, but this is the now Cancer Centre, which opened in 2005. This is the Centre for Cancer Research and Cell Biology. And these two buildings now really integrate very well. Richard is now the head of medical oncology, basically working between both of these buildings with the whole team. This is the inside of CCRCB. But importantly, they bring that theme that I actually heralded before of integrating cancer discovery in terms of clinical and scientific research and how that impacts on patients. And this indeed is now the next generation development of that, which will open in 2015. And this building will then, this is the current CCRCB, this is the new Centre for Experimental Medicine along with that. And together, these two buildings will house 700 scientists. So you can see the impact of that. Beyond that, there's now a health sciences campus. So once this clinical centre was built, CCRCB was opened 2 years later, and now there's a whole research campus, which is in other areas like infection immunity, and actually by 2015, the whole campus here will have close to 1,000 scientists. There's a whole Wellcome Clinical Research Facility now built here. We're currently talking about building an experimental hospital on this campus, but the power of focusing on the patient, doing the right thing, can really suddenly transform not just things for the cancer patient, but actually for patients well beyond that. And this really has galvanized the whole medical community, not just in Belfast or in Northern Ireland, but also, I think it's fair to say, right across the whole island. I suppose one of the proudest moments I've had in terms of an external acclamation of this outside of today was when Her Majesty the Queen last year awarded us her Diamond Jubilee Award. There were 20 awards given out in the UK, and this is a picture of myself and the Vice-Chancellor meeting herself and the Duke of Edinburgh, and that was because this programme had led to Northern Ireland having the best outcomes for cancer survival in the United Kingdom. So in a period of 12 years. So that was a very proud moment for that to be acknowledged in that way. But most importantly, Professor Murphy got his Irish citizenship back for his family. This was a wonderful moment where, again, Tom Moran behind the scenes, but I was honoured to be there, as was Iseult. Bertie Ahern was the Prime Minister, but Marty had been asked by his father to go and retrieve his family heritage and their Irish citizenship. And Bertie Ahern personally came along to present that to Marty and to Anne in Galway one summer about 8 years ago. So you see, when you actually do the right thing by patients, look at what can happen. Even Murphy can get his Irish citizenship back. So let's get on to the science and what we really do now. So back when I was working on thymidylate synthase, very important, but everybody quite rightly was saying, well, Paddy, it's really nice work, but so what? And that's what I've been known as, actually, in the Cancer Centre, as Professor So What. And the reason for that was, well, what were we going to do? I mean, no matter how much work that Fritz or Peter Dannenberg or I did, the clinical community wasn't going to move away from one drug to nothing. And it does sound very strange, but I remember a conversation with Bruce around about 1994, 95, talking about, you know, where were we going to go in terms of treating colorectal cancer. Oxaliplatin wasn't really a very, well, actually, by the way, oxaliplatin isn't an active drug in colorectal cancer, not unless you put it together with 5-FU. First studies with oxaliplatin were not, it wasn't active. It's actually only when you combine it in the thymidylate state that it actually starts to work. But importantly, it just shows you the transformation that we've now had. And of course, I took the philosophy of, well, let's go and try and, you know, build a way of targeting the right patients. So building on the themes that we had actually come forward with. When I pulled this slide together in 2006, there were 64 drugs. And this is an old slide, in development in colorectal cancer. We were still using old paradigms, and by the way, they just weren't working. And in some ways, they're not working now, but you can see from what Tim has said today that people have now really started to search for what's the best way to go forward. So We took an approach as well, let's try and understand how we get at targeting the genetics and then move towards targeting the biology around pathways. But the problem at the time was that we recognized that just looking at P53 mutations or KRAS mutations on their own really wasn't going to be enough. There was no one marker that was likely to actually lead us anywhere. And back in 1999, I had started working with Affymetrix. I'd actually been out visiting UCSF, and I actually went to visit Affy, which were just situated very close to the airport in San Francisco, and they had developed the 9K array. So Paul Harkin had actually done some prototype work when he was at the Farber, or at MGH, working with this. So we started to work with the 9K array, playing around with tissues to see what sort of things we could actually do. But at the same time, we said, you know, what these arrays are doing is actually taking data sets from the Human Genome Database and actually putting those transcripts onto the array platforms. They're not really thinking about things in terms of a disease context. And so what we started doing was actually a very prolonged exercise, because then doing high throughput sequencing, building libraries, so this is 2002, 2003, that sort of time frame was actually really of a huge volume of work. So we started building libraries for colorectal cancer, for breast cancer, lung cancer. And what we did was we took a 3' sequencing approach to this, very deliberately. We didn't take a 5', which would have been the more common way to do that. Yes, we were trying to build a library of information, but a lot of that wasn't certain contexts out there, but what wasn't there was that done in a tissue context. And what was novel about this concept was we were trying to do it in a somatic tissue context and then apply it in a certain way. And the important thing was for us, we wanted to try to optimize our ability to try and deliver this in paraffin-embedded tissue, because that's the way patients get treated, that's the way tissues get stored, and that's how we're going to actually try and move ahead. And so we did that successfully. And these are now all on Affy platforms, and indeed the Accelerate, which Richard has helped design, synthesizes all of this information, and actually is now, that's all captured on the Accelerate, which is Affymetrix' now preferred platform for doing discovery as a tool in paraffin-embedded samples. But of course, we've gone on then to use these platforms and the Accelerate, as was outlined by Richard, to really do and drive a lot of our discovery. And for us, one of the big challenges was, as has been outlined today by numerous speakers, and particularly by Tim, is that we can no longer really accept the mantra that we can go from metastatic disease to primary tissue easily, or adjuvant setting in terms of treatment. And while the metastatic disease setting will tell us an awful lot, it isn't actually translating. So we were being slowed down to some extent by not being able to actually take the information from metastatic disease into the early adjuvant setting. And the reason for that is we don't really know how that epithelial setting that's established in the metastatic context really relates to the primary even still. But what we have begun to learn is that it's very different. And therefore, a new approach was needed to really begin to build a relevant understanding of the genetic background alongside the somatic phenotype. And that's what we've all been talking about. Data which was published by Richard back in JCO in 2011 looked at, and this was work, by the way, that took 6.5 years to do. We came forward with probably at least 4 or 5 different signatures across 6 years, none of which we've ever published, very deliberately, because we couldn't stand over them. We couldn't stand over them in terms of a validation. We published a signature based on the DSA platform for colorectal cancer. That actually showed that we had a number of molecular subtypes. This is Richard's publication from JCO 2 years ago, showing that we had a signature that was able to predict recurrence with a hazard ratio of 2.6 for stage 2. And indeed, not published yet, but this has just been validated in the last 2 weeks by CalGB This signature is now licensed to Precision Therapeutics, who will no doubt further develop it. But importantly, what it began to show us was that within stage 2, unlike what Jean-Paul was showing, we actually were finding 4 different subsets within stage 2. And so very much for us was saying, well, yeah, absolutely, there are now different subsets of colorectal cancer, and we're going to have to begin to mine and understand these in much more detail. And that's indeed what we've gone on to do now. This is a work in progress with 10 different labs across Europe and the US, where we're taking both a sequencing approach of fresh tissue, not of paraffin tissue, using a high throughput sequencing to define the mutational landscape. We're also linking to do a supervised classification, looking at the transcriptome profile to try and validate whether there are 3 or 4 profiles that are relevant, linking those back to the mutational screen that we find, and we're doing this in collaboration with the Sanger Centre as well as with Oxford. And we're also going to use connectivity mapping to link into the Sanger screen cell line sensitivity assays with all this information to try and come forward with a map of how particular transcriptional profiles and mutational maps actually link to sensitivity. And this work, which is funded by Cancer Research UK, is now well underway, and it'll probably be another 2 years before we really begin to report some of the initial important information on this. The other big thing that's already been talked about is, of course, the relevance of KRAS. KRAS was important for the colorectal field only because it really shifted the thinking and created a new paradigm where EGFR inhibitors should only be used in those colorectal cancers that are EGFR wild type. That came about, as Tim has shown you, by serendipity. In actual fact, it came about because a drug was about to get canned in Europe, and they went back retrospectively, Amgen did, to try and rescue panitumumab, and that thankfully showed a very important association. And Rafael D'Amato's paper was very important in moving the field forward. So what we've taken the approach then is trying to define targets that may be relevant in a KRAS mutant context. And so what we've done over the last couple of years is we've begun to build a large data set of both PDX model, xenograph models, primary tumors, and also then searching the public databases in terms of trying to look at those transcriptional targets that appear to be highly relevant and differentially expressed across KRAS wild type and mutant contexts using cell lines and all of these other things. And then what we've done is we've taken those through a number of screens, primary, secondary, validation, and using metacore analysis. So we've taken out nodal points on each of these transcriptome maps and actually then done our screens through the primary, secondary, and then tertiary validation of these. The one I'm going to highlight today is the JAK1/2 STAT3 pathway, which has been led by Sandra van Scheebroek in her work and its functional validation. Where, so the JAK-STAT pathway, when STAT gets activated, it transcriptionally regulates a number of important genes shown here, but regulates cell survival pathways, migration and proliferation, depending on the contextual relationship. We've also been able to show, and I'll come to this later, that ADAM17, a SHEDase that my lab has been working on for several years, actually regulates some of these key receptors, including EGFR, by creating ligands that promiscuously actually activate these pathways under certain circumstances, and in particular that this is regulated by ADAM17, is regulated by KRAS, but also induced heavily by chemotherapy. This actually shows the combination of JAK2 and MEK inhibition, just here, in the HCT116 cell line, which is mutant to RAS, versus the HKH2, which is wild type. And as you will see, the addition of JAK1/2 inhibition to MEK inhibition significantly synergizes this. And you see exactly the same thing then in our in vivo models. This is just the HCT116 models, where you see very significant synergy in terms of those models, in terms of knocking out both MEK but also JAK1/2 in the RAS mutant model. So very much suggesting that in this model, this may be very important. And you can see over here that we've not only knocked out ERK, but we've also knocked out the inhibition of phospho-STAT3 and its activation. We've gone on to further look at this and asked the question, well, what could be regulating JAK induction here? What's the upstream regulator and the receptor that's involved? And this basically shows that c-Met, which was one of the candidates, when we knock out c-Met using crizotinib in conjunction with MEK inhibition using the AZD6244, we actually deactivate this whole pathway, which is activated, and also downstream activation of STAT is also deactivated. So very much pointing to c-Met as being a key regulator of this JAK-STAT pathway in the RAS mutant context. We've actually then looked to see, is ADAM17 involved in this in some way, in actually regulating c-Met and how it is activated. And though I'm not taking you through a lot of the detail because of time, we actually have found that indeed it is. If you actually just look over here, this is when we induce ADAM17 activity. And what you see is this is the level, the x-axis is the level of soluble c-Met. And you get a dramatic induction of the level of soluble c-Met when you actually activate ADAM17. And you only get minor decrease of that when you then expose these to a MEK inhibitor. And you can see again, here's the MEK inhibitor on its own. But if you actually induce the MET receptor with ADAM17 and its activation, those xenografts are rescued and they actually go ahead. We've then gone on to test this whole hypothesis by using a number of MET inhibitors, including crizotinib in conjunction with MEK inhibition. And again, this line here is the combination of MET and MEK inhibition. And as you can see, we get very significant synergy and shutdown of these tumours with that combination. This has now resulted in a very important trial, which Mercuric, which is now funded through the FP7 programme. There are 8 centres across Europe, including Tim and his team in Oxford, where just as Tim had described for other combinations, we're doing a pilot now to look at the combination of MEK inhibition and MET inhibition. These are Pfizer compounds, and Pfizer is a collaborator in this alongside the Framework 7 programme. And we have a first phase, which is looking at the dose, but we're biopsying patients both pre and post in doing this. And then the phase 2 will stratify patients based on KRAS wild type, c-Met expression, or KRAS mutant colorectal cancer. And then we will have 2 standard arms in combining MET-MEK inhibition or chemotherapy. Now, the importance of this is that if this trial actually really shows us something and real activity in this defined population, we will then take that to Focus 4, which Tim has given a very eloquent and deliberate expose of. Because this actually, in a sense, is probably for me one of the most exciting developments in GI cancers, possibly in cancer in general, because this type of trial now facilitates us really beginning to pilot things in a very deliberate way and also come up with schedules and regimens that are about targeted or stratified populations and that may have real impact. So hopefully, if this MEK inhibition schedule actually works, we will then actually be able to come to Tim and say, Tim, have a look at this data as a proposal for another arm as part of targeting RAS mutant tumours going forward. So just to wind up then, I mean, as you can see, my career has come full circle in some ways, starting off, as a fellow at NIH. But it has always been about trying to accelerate the discovery through integrated clinical and scientific research and about improving the lot of patients. And that has followed all of this journey, whether it's working with NIH, whether it's working with CRUK, or indeed more laterally with MRC. And it's all about partnerships between universities, biotechs, the NHS industry, and leveraging our unique strengths. And every institution, no matter how small or how big you are, you don't have it all. You can't have it all. You will always have areas that you need to collaborate with, and you'll need to find those partners. So in summary, I suppose the translational journey for me, Bethesda to Belfast, the journey that I started with Bruce, with Eli, with Marty and Bob and many other people that have come along the way, is about and has been about really building a comprehensive care system, not really a building, but one that really cares about discovery and application and how those impact people and patients. It's been about integrating and collaborating scientifically in a way that's clinically relevant. And that's the so what question, by the way. It's also been, however, about innovation. You know, thinking out of the box, do it differently. Let's see what it looks like. And of course, that's all about taking your ideas, making them commercial as well, because we need that. We need that type of innovation going on. And innovation must underpin all of our work in personalized medicine, because I can tell you, if we come back here in 10 years' time talking about personalized medicine, we'll be talking about all the mistakes we made and why we must do it slightly differently. But most of all, I think I want to leave you with the importance of mentorship. I'm still being mentored, by the way. I badly need it. Work in progress, you know, that's the way it's always going to be. But also teamwork, friendship, and kinship, because increasingly in life, as I get further down that road, it's those things that I want to go and work on. It's with those people. It's those people that are there who are actually selflessly trying to make a difference for patients and for society. And finally, I just want to acknowledge a few people. Some are here in the room. Paul Harkin was my first recruit. Paul had been at MGH, worked with Dan Haber, and Paul came back to work with me in 1997. And I have to say he's been a tremendous friend, but also a tremendous collaborator. Richard and Alton McDermott, who's now at the Sanger, were my two first clinical fellows. And they were tremendous role models to have, because they brought that compassion and commitment to what they did. David Waugh was my second external scientific recruit and is now the director of CCRCB, and also a very successful scientist in his own right. And Richard Wilson was in training in Belfast when I arrived, and sent him off to the NCI, and Richard leads our whole clinical trial program. I also just want to acknowledge Dan Longley, who started as a postdoc with me, is now an associate professor in the university, leads all the death signaling and flip work. Sandra van Scheebroek, who came from Belgium to do her PhD with me and is now in her second CRUK clinician scientist post. Mark Lawler, Mark was a very important player in the NCI All-Ireland rollout. He actually was the person who took it on in the south of Ireland and has now come to join me working in Belfast. And Manuel Salto Teles, who recently joined us from Singapore, has developed the whole molecular diagnostics outfit. So I'd like to thank all of them, my Mercuric partners, and all of the groups that are currently working with us on the CRC taxonomy project, and also all of the people who funded my work over the course of the last 15, 20 years. Thank you very much indeed.
So thank you very much, and congratulations from all of us from FMC. I have also for you the small present.
A book from Amsterdam. Thank you, everyone.
Thank you. In the matter of time, we don't have time for questions. I just want to close this meeting today with a general remark that I learned a lot, and I hope you did too. We had a very exciting program, I guess, and all the speakers I want to thank for their sharing their thoughts and the fruitful discussions. I want to thank the STO, especially Anne Murphy, Martin Murphy, Gabby Cruz, and I want to give the last thank you to the two most important persons for the day to make it possible, and that's actually for Joke van Diemen and Mariette Labot, because if they weren't here, we would not have been here, and the speakers would not have been here. So I would like to ask you to give them a great applause for organizing everything.